This is part 3 of chapter 1. This segment should take us through the end of chapter 1. We're going to start this section by talking about Louis Pasteur and the germ theory of disease. It was Pasteur that developed that idea that microorganisms were the cause of disease, but it was Robert Koch that was able to actually be the first individual to definitively link a particular microorganism with a disease. That disease was anthrax. He beat Pasteur to the punch by demonstrating that the organism Bacillus anthracis caused that condition, anthrax. This was kind of a big deal. Pasteur was pretty unhappy about that particular outcome, as he had been attempting to show something along those same lines as well. Robert Koch did a lot of important work in the laboratory, and it has relevance to our work today. In fact, a researcher working in his lab figured out how to add agar instead of gelatin to media so they could better grow and examine colonies of microorganisms on a petri plate. He also developed what are known as Koch's postulates. This won't be the only time we speak of them, but we will introduce them here in a slide or two. It was the process he used to show a specific organism caused a specific disease. And while this process has lots of relevance even today, he also came up with a lot of other firsts. Besides anthrax, he also showed that another organism was the cause of tuberculosis, the bacterium Mycobacterium tuberculosis. Thus, he was able to link 2 different diseases to 2 specific organisms. He was also the individual that was able to demonstrate staining techniques for microorganisms, something that we do all the time in the microbiology lab. He also took the first photograph called a micrograph, so a photo of an organism under the microscope. He also took the first photograph showing bacteria in a diseased infected tissue. He also came up with techniques to estimate the number of bacteria. He used steam to sterilize media. He used aseptic transfer processes. His lab crew came up with the agar and media in petri dishes. Very many routine procedures in the microbiology lab have origins in the work of Robert Koch and his lab. The next image is about the different colonies that you might see on a plate. When you look at this particular picture, you see all kinds of different shapes and colors, different sizes of colonies. These are all indicative of different types of microorganisms. You might see something like this when you do an experiment where you're testing what's in the environment. A moment ago, I introduced Koch's postulates. This is that process he used and is still used today to link an organism to a disease. Basically, Koch's postulate said that in order to definitively show that a microorganism caused a particular disease, you had to first find it in every case of disease. You had to then be able to isolate the organism in the laboratory to work with it in pure culture. Once grown in the lab, you would reintroduce that organism to a susceptible host, an animal that could be infected, and then that animal should become sick with the same condition, that same disease. From that sick animal, you could then re-isolate the same organism. This process was really an important one that Koch worked out to show that a specific organism was linked to a specific disease. We'll come back to Koch's postulates later in the semester when we're talking about infectious diseases, but these are very important ideas that seem fairly straightforward now, but they were pretty groundbreaking at the time. It was the first time that scientists were able to definitively show that a particular organism was the agent that was causing the infection. Another really important procedure was the discovery of the gram stain. This is a fundamental procedure for microbiology and even in diagnostics. You can see an image of a gram stain right here in this micrograph. Can you see these little pink cells and the little purple cells? These are gram-negative when they stain pink and gram-positive when they stain purple. Hans Christian Gram was the microbiologist that came up with this particular procedure. You will get to perform gram stains in lab and even be scored on the quality of your stain. It is that fundamental to microbiology. It's extremely important as a step to identifying organisms. Of course, since we're talking about this procedure in this section, this discovery was also something that happened during this golden age of microbiology. Returning to the question regarding disease transmission and disease prevention, there were 6 important individuals that did work to help promote the ideas concerning protecting ourselves from infectious microorganisms. So who are they? The first is Ignaz Semmelweis. Semmelweis was the first individual that put forth the idea that, medical professionals, doctors, nurses should be washing their hands in between patients. Seems pretty obvious and is very routine nowadays, but you can imagine a time when they knew little about microbes. Hand washing was not an obvious necessity for disease prevention. But Semmelweis suspected a link between the lack of hand washing and the transmission of puerperal fever, which is sometimes called childbed fever. So the idea was, when he was working in the hospital, he witnessed physicians going from the morgue, where they were doing autopsies on diseased patients, trying to figure out why they died, straight into the labor and delivery without washing their hands. Something that is horrifying to think of nowadays. But remember, there was a general lack of understanding regarding the transmission of disease back then. Semmelweis, he was intuitive, and he knew that was just not right. You were going from people that died of infections into a labor and delivery. And then these women were dying of what was said to be infections related to childbirth. He knew that this practice was a problem. He was trying to tell doctors to wash their hands. Unfortunately, that didn't go over very well. These professionals, used to being regarded in high esteem as lifesavers, were being told they could cause infection and death. They thought, what are you saying? That we're the ones causing the death? They just couldn't believe it. They ridiculed Semmelweis incessantly. Think of a bad case of cyberbullying, but, in the old days. They actually caused poor Semmelweis to essentially lose his mind from the pressure of the ridicule he was subjected to. Then, in an ugly twist of fate, he actually ended up dying from the same organism that causes puerperal fever. Next, Joseph Lister came along. He had recognized the value of Semmelweis's work, and his disposition was much more resilient. Like, he was kind of a stubborn guy, very, pushy, let's say. He absolutely insisted that the people working under him in the hospital use antiseptics to wash their hands very thoroughly. Of course, as you might expect, infection rates in the hospital went way down because of good hygiene. He promoted his good data. Eventually, the ideas about aseptic practices started to gain some traction over work like this. Another important individual was female. Historically important females are not as abundant as the males for many societal reasons, but she's one of them. A strong-willed nurse, she had a great impact on the hygiene of hospitals. You may have heard of her, Florence Nightingale. She was a nurse working during the Crimean War, and she recognized that the filthy conditions that the soldiers were subjected to coming into the hospital was contributing to the infection rate. She became absolutely insistent at implementing good hygiene and keeping things clean and sanitary. Of course, this helped bring the infection rates down. Not surprisingly, she has been well regarded by history for instituting those changes in a hospital setting. Next, your text speaks of John Snow. We will learn more about him later in the semester, but again, he was an important individual for infection control and epidemiology. He linked an infection in London to the water supply. He used data to convince authorities to shut down a particular water pump, and by doing so, the cholera epidemic that was raging was brought under control. Because of this work, John Snow is known as the father of epidemiology. While we still will talk more about him in chapter 14, if you like historical stories, read the book The Ghost Map. Our library has a copy. It's a really interesting read about the story of the origins of epidemiology. It talks about his experience and how he tried to solve the mystery of this outbreak. I enjoyed it a lot, and I learned a few things about historical London along the way. The next individual your text discusses is Edward Jenner. You may have recognized this name already. He came up with the first real vaccine, which was against smallpox. He found that the related infection, cowpox, could serve to protect against the more devastating smallpox. The term vaccine is referring to vaca, or cow, as a nod to the origin of the first vaccine using cowpox. We'll talk more about Jenner later when we get to discussing vaccines, But since the discovery of the ability to protect against infection was so important, he is specifically highlighted in this section along with the other historically important people. The sixth individual discussed in your text is Paul Ehrlich. Ehrlich came up with the idea that there should be chemicals that would be harmful to microorganisms but not the infected host. He coined the term magic bullet to describe this concept of targeted therapy. He set out to find something that would kill just the organism and not the host. That's the basis of chemotherapy and antimicrobials, all of that. He searched long and hard for these magic bullets, but he really didn't come up with a very good one. His discoveries might kill microbes, but they weren't so good for the infected person either. We'll talk more about that in a later section, but importantly, he had the idea, and that idea was able to stick. Here we go. I'm going to give you a little quiz, just to see if you're awake. Are you still listening to me? Which scientists was the one that first hypothesized that medical personnel will infect patients with pathogens? We have 4 choices: Edward Jenner, Joseph Lister, John Snow, or Ignaz Semmelweis. Which? Gotta guess. Well, if you said it was Semmelweis, you were listening. Ding, ding, ding. So you got it right. Choice D is correct. Remember, he was the first to realize that hand washing was really important, something that we are still reminded daily about right now. On this slide, there are terms that I'm not going to go through, but I'm going to direct you to go look through table 1.3 in your textbook. It's going to go through some of the terminology of the fields that are related to microbiology, and it would be wise to expand your vocabulary and provide yourself some of this knowledge, especially if you're not that familiar with it. Most of you plan to enter the health-related careers, and these terms will be used. You want to know what's being referred to when you see and hear these different terms. All right, now we're going to skip to some of the basic chemical reactions of life. Biochemistry is importantly linked to microbiology. It really all started with Pasteur's work on fermentation and understanding that yeast were the ones that were directing the work. Buchner showed that enzymes alone could do the work, but there are other individuals like Kluber and Van Niel that used microbes as the model systems for biochemical reactions. Biochemistry is really linked to microbes. Microbes are the best biochemists. They really are. There is such a huge diversity, so many different types of microbes living in every nook and cranny on this planet. If you look into their collective abilities, they're really quite amazing. Different species are able to do very interesting things with the chemicals in the environment, and we're really just beginning to explore that huge potential out there. You might be wondering why that biochemical diversity is important. Well, what's the application? There are many. The design of herbicides and pesticides, diagnosis of illnesses, understanding our own metabolism and patient responses, metabolic diseases, treatments, drug designs, and more. There are so many things that microbes can do, and we're still learning how to harness their abilities for the good of humans and the earth itself. We'll be spending more time in a later chapter looking at how genes work, but I just wanted to make a point that understanding how genes work relates again to microbiology. Microbes have been and still are the model system for understanding genetics because they have fast replication rate and the basis of life is the same. The basis of the genetic instructions are the same whether you're a prokaryote or a eukaryotic organism. Microorganisms are model systems for understanding the advances in genetics. In fact, the greatest advance that has hit genetics in a really long time comes from bacteria. You may have heard of CRISPR. Hopefully we'll be able to talk a little bit more about CRISPR later. This particular system is going to be revolutionary in treating diseases, and it has so many other scientific applications. I feel quite confident that in your lifetime, you will see huge advances in gene therapy because of that discovery. This genetic system originates as a bacterial immune system. So again, microbes are responsible for huge advances in genetics. These fields, like genetics and molecular biology, they all have origins in microbiology. We're going to wrap up chapter 1 considering some environmental applications. Have you heard of bioremediation? That's when we can detoxify polluted environments using microorganisms, or at least other living organisms, to deal with the contamination. Microbes, as I mentioned, have a huge and diverse biochemical ability. If you put a chemical out there, there's probably a microbe capable of breaking it down and transforming it into something less harmful. That's the concept behind bioremediation. Microorganisms are responsible for recycling all kinds of chemicals or nutrients. They are at each end of the cycles for pretty much every element important to life, like carbon, nitrogen, and sulfur. Of course, microbes are important for generating oxygen. I know you all learned about plants making oxygen for our environment, but microbes are clearly responsible for at least half of the oxygen out there. All of those algal microorganisms out there, the phytoplankton that photosynthesize, they produce a lot of oxygen for our biosphere. All right, so when it comes about defending diseases, later in the semester, and this is just a little preview, we'll be looking at some of these ideas or fields: serology, immunology, and chemotherapy. All of these are, again, related to microbial systems and microbial fields, or I wouldn't be mentioning them here right now in this introductory chapter. This little figure that's shown here, maybe it's something that looks a little familiar to you. You might have seen something like this in another textbook. This is sort of a smear on the agar of staph. And you see this mold growing right there? Well, what you have is a clear zone in between the mold and the bacteria. This is called a zone of inhibition. Clearly, the mold is making something that is inhibiting the bacteria. We now know that molds, specific species, can make penicillin that inhibits staphylococci, or at least most wild-type staphylococci, as well as some other bacteria. Other types of molds and different microorganisms produce different antimicrobials that inhibit different microbes. This is to eliminate the competition that wants the same resources they do. Microbial weapons. More on these later. So I'd like to close out this particular chapter with one review question and a micro-themed joke. I like micro-themed jokes and memes, so you can send them my way anytime when you come across them. Okay, here we go. Question: who's responsible for building the first simple microscope? This is from the beginning of the chapter. Think back. Was it Isaac Newton, the Royal Society of London, Antony van Leeuwenhoek, or Louis Pasteur? Hopefully, you recalled it was Antony van Leeuwenhoek. All right, here comes my corny micro joke. What was said of the microbiologist who traveled the world? Why, they appreciated many cultures, of course. All right, I'm out. This is the last chapter of chapter 1. I'll see you in chapter 2. Here's hoping you have a fabulous day.